Hello everyone. Today's topic is the cell cycle. Now, what is the cell cycle? The cell cycle is a series of events that replicate DNA and cause cell division. It is divided into two phases, interphase and mitosis. Interphase is divided into G1, S, and G2. G1 stands for gap 1, S for synthesis, and G2 for gap 2. In mitosis, the cell divides into two daughter cells, basically two identical cells. The cell cycle is strictly controlled to prevent cell damage or abnormal DNA being passed on. In case there is damage, since mistakes can occur, apoptosis commences. Apoptosis is the process of controlled cell death to prevent any further damage. Now, interphase is the interval between each cell division, where the cell prepares for the next division, so mitosis. And as previously mentioned, there are three phases, G1, S, and G2. G1 can take several hours to months, so let's write that down. G1 can take hours to months, depending on the type of cell. This phase occurs after mitosis, and there is synthesis of RNA, so let's write that down. RNA, proteins, and cell organelles. Now, the cell grows since the end result will be two identical daughter cells, so it has to have more matter to be able to replicate properly. Also, nucleotide excision repair is done. So nucleotide excision repair. And there's also a G1 checkpoint right before entering the S phase, just to make sure that everything is being copied properly, everything is done properly, and there are no mistakes being done. So let's write that down so we don't forget it. G1 checkpoint. Now, the S phase. The S phase takes around 8 hours long to complete, so let's write that down. 8 hours, and during this phase, DNA replication results in two sister chromatids per chromosome. So what this means is that there are going to be two copies of each chromosome. Remember, the chromosome usually looks like this. Well, is being represented in images like this. And what we will normally see in the other drawings is that it's going to look like this. So two copies of each chromosome. Let's just erase that. And so what happens during the S phase? There is synthesis of proteins needed for DNA packaging. So let's write that down. Synthesis of proteins for DNA packaging. And there's also mismatch repair. So we don't want those. We don't want the copy of the chromosome attaching itself to another chromosome. We have to make sure that they're all properly attached with their correct chromosome. And once the cell commences S phase, it must complete the whole process. There is no turning back to the G1 phase. Now for G2, It takes around 2 to 5 hours to complete. And where further synthesis of protein is done. Also, errors in the DNA replication is repaired. And there is also a G2 checkpoint right before entering mitosis, like we said, to repair any errors that have been made. And so we don't want a faulty cell being replicated, since that can cause a lot of damage in the body, like for example, cancer. G2 checkpoint. Now, as you also see, there is a G0, as you can see right here, and it is not inside of the cycle, as in this cycle it is separated. So what is this G0? G0 is also known as the resting phase. Here, let's write this down. And this is a phase that the cell enters after exiting the G1 phase. The cells are differentiated and have certain functions and are considered to be going under cell division. There are a few cells that re-enter the G1 phase when exposed to certain conditions, like if it needs to replicate after some damage has been done to an organ, or to fix any damage done. But most mature cells or tissues are in the resting phase. There are also some types of cells, like neurons, for example, that do not replicate ever again once they are formed, so they are always constantly in G0. Now, for mitosis, or the M phase, there are five phases inside of mitosis. Now, mitosis is the process in which cell distributes the DNA to the daughter cells, and it lasts around 1 hour. So let's write that down. Let's write it over here. Around 1 hour. And like I said, there are 5 different phases, each very important and have key roles. So first is prophase, which is this one right here. Now, prophase is the first phase in mitosis. Here, the chromosomes condensate and the centrosomes separate. The centrosome is the point of origin of the mitotic spindle. The mitotic spindle being these lines that help the chromosomes align for later phases. So there, in some texts, in some books, they consider the prophase being 2 phases, so prophase and late prophase. So here we'll be saying there's the prophase and the late prophase. So in the first image, in this one, is where the chromosome condensation, and it starts preparing itself for the spindles to be able to align and continue to the next phase. So let's write this down. So chromosome condensation. And the centrosome separate. So the centrosome, sorry, I forgot to mention this. The centrosome would be right here. I tried to draw. You can see it over here more clearly. There's that white point or line is where the centrosome would be. and those are at the two ends of the cell. And there's also a matrix surrounding all of the spindles, and microtubules would be this line, where the one they're emerging from the centrosomes. Now the next phase would be metaphase, which is this one right here. Metaphase is, hold on. Metaphase would be where the chromosomes are super condensed and aligned in the center of the cell. So let's write that down. And the alignment. Then there is anaphase, which would be this one right here. Anaphase is the separation of the sister chromatids. So as you can see right here, as we had mentioned before, these these images, each one, like this one and then this one, are two copies of the same chromosome, like I had mentioned before. So here, each copy is being pulled into different ends or different poles of the cell, so that they can they can pinch, so the cell can pinch and divide into two equal cells, or two identical cells, sorry. So in anaphase, there's the separation of the sister chromatids, since the cohesion, what attached each chromosome, is dissolved by the enzyme separase. So let's write this down. So separation of sister chromatids. And cohesion dissolved by enzyme separase. And also the cell is elongated, so it can start pinching in the middle in the next phase, which is telophase. So telophase is where the chromosomes decondensation occurs. So it can start look so it can so it's not as compressed as it was before. It can be a normal cell. The disintegration of the mitotic spindle. So these spindles that attach to the chromatids are dissolved, and a new nuclear membrane is formed, represented by this blue circle. And the cell body divides at the center of the ribosomal divides at the center, and ribosomal or rRNA is synthesized. So this is where it pinches and it separates, and the ribosomal RNA, or as sometimes addressed in books, rRNA, is synthesized. Let's write all of this down. Chromosome decondensation. Disintegration of mitotic spindle. The new nuclear membrane formed. The cell body divides at the center. And the ribosomal or rRNA is synthesized. So rRNA synthesis. And finally, we have cytokinesis. Cytokinesis is the division of the cell cytoplasm into two new cell bodies. So let's write this down. So division of cells into two new cell bodies. And let's just mention a few things about this. It usually begins at the end of anaphase, so it starts right after this into the process of telophase. It usually occurs at the same time as telophase. So the cytoplasm and the cell organelles that we said were copied to have identical on both cells are distributed between the two new cells, as they both will need organelles to properly function. And each of the new cells contains one copy of the sister chromatids. As we mentioned, these are identical. So let's write all of this down. Cytoplasm and organelles evenly distributed, and the cells contain one copy of chromatids. And that is the cell cycle. Please like the video and subscribe to our channel to support our channel. If you have any questions or feedback, please leave a comment so we may answer your questions and help make our videos better. Thank you.